Right. So, yes, without wasting much time, let's go for our today's session on what is called as your plant growth. Right. So, as you know that this is a live class for people who have been struggling with biology. So, we'll be having a very quick one by one, line by line review of this plant growth, because this plant, just give me a moment. Review of this plant growth, because this plant, just give me a moment. Yes, sorry, I'm there. So we actually know that plant physiology. If you look at the last few papers which I went through the NEET, there were every unit wise there is around 10 questions at least, right? There are 10 question units in biology, so you can expect around 12 to 13 questions per unit, right? So as far as plant physiology is concerned, म्हणजे मला यावेळेस जरा का असं वाटतंय. Right. So, as I told that we'll be talking about this particular phenomenon of what is called as the plant growth. So, I told that one by one, I was talking about the plant physiology, and what I told that in this plant physiology, you can expect around 12, 13 questions, 15 questions. Sometimes in 2022, there are around 13, 14 questions on this plant physiology. I think approximately 15. So, unit wise, you're going to have that many questions. But now, here you see, you have omitted that mineral nutrition, you have omitted that transport in plants. So what remains is only three: the photosynthesis, respiration, and the plant growth. So you may have maybe around 4 to 5 questions maybe, we don't know, on each particular chapter of this plant physiology. So talking about plant growth, so let us go one by one, the line, the words which are given in NCERT related to this plant growth. So plant growth, as I said, that different organs, they are showing growth in different patterns, like the stem which is there, the apexes which are there, the stem apex or the root apex, right? They are showing unlimited growth, the height is increasing. But then other parts like your leaves, your flowers, your fruits, right, they are showing a limited, what you call as, growth, right? So also you'll find that some parts they appear and some parts they fall and they appear again, right? So the plant overall, the growth patterns which are there, they are showing a different what you call as a pattern in different, what you call as, organisms. We all started the cell life from this, what is called as a zygote, right? So the life of a plant, it started from the zygote, and from this zygote there was growth, there was development, and then you know that famous equation that development is the sum of growth as well as differentiation, right? So sum of growth and differentiation, right, that is what is called as your development. development. So there is growth, there is differentiation, there is a very precise, they've used the word precise, very orderly events which are taking place in the growth of the plant. So you have roots coming out, you have stem coming out, you have leaves coming out, then you have flowers, you have branches, you have fruits, then you have seeds, and then the death, right? So there is a very orderly events, a very precise series of events which is happening place, and here you have the first step in the growth of the plant, that is your germination of your seed. So the first step with which the growth of the plant it starts is the germination of your seed, which will occur only when the conditions are favorable. So if it is having a proper temperature, if it is having a proper moisture, if it is having proper nutrients, only then the seeds they will germinate. If the conditions are unfavorable, right, so you will find that in these unfavorable conditions the plant the seed actually will not grow. It will go into the period of what is called as a dormancy, where the seed it will not germinate, it will go rest, and there will be a suspended growth, what you say, right? So the plant growth which is there, it will be suspended, and whenever the favorable conditions return, remember the metabolic activity it increases. So there is an increase in the metabolic activity, there is an increase in the growth, and we know that this occurs due to both the factors, the intrinsic factors and the extrinsic factors. Both are actually responsible for the growth of the plant, right? So the internal factors, right, and the external factors, the light, temperature, and all, they are external factors. The internal, the what you call as the genetic factors, or you can say the plant growth hormones which are there, they are the internal factors. So all these are actually going to affect the growth of the plant. So growth basically, you have to remember, is a fundamental process which is taking place in all living organisms. It is conspicuous. Sometimes you feel like these words are not very important, काय तेच कॉन्स्पिक्युअस, बट कधी कधी मागच्या नीट मध्ये ते विचारलं होतं की टेरिडोफाइटचा जो प्रोथालस आहे तो कॉन्स्पिक्युअस आहे का इनकॉन्स्पिक्युअस आहे. सो आय एम नॉट व्हेरी शुअर व्हॉट दे कॅन आस्क, बट देन स्टिल यू हैव टू रिमेंबर इट इज अ फंडामेंटल प्रोसेस, इट इज अ कॉन्स्पिक्युअस प्रोसेस, it is an irreversible process, and it is a permanent increase in what you call as size, shape, length, whatever you call, right? So it may be increase in the size of the organ, the parts, or even that of an individual cell, right? And that occurs because of what is called as metabolism. Remember that this metabolism is the sum of both anabolism plus catabolism. दोन्ही होणार आहे. It is not only synthesis. Sometimes you feel like growth म्हणजे anabolism होत असेल, नाही. So it may be anabolism or it may be catabolism. दोन्ही पैकी नाही, दोन्ही मिळून काय घडून आणतात तुमची ग्रोथ. आय एम नॉट गोइंग इनटू एक्सप्लेनेशन, कारण मला एका तासाच्या आत हा चॅप्टर संपवायचा आहे. तर म्हणजे वी आर नॉट गोइंग टू गो इन टू द डिटेल्स ऑफ द कॉन्सेप्ट्स, बट द वर्ड्स, राईट? सो वर्ड इम्पॉर्टंट हियर इज दॅट बोथ anabolism and catabolism, they are responsible for growth and they are going to require energy. तर या सगळ्यासाठी त्यांना काय लागणार आहे? एनर्जी. सो इफ यू हॅव अ लीफ एक्सपांडिंग, राईट, दॅट इज ऑल्सो द एक्झाम्पल ऑफ व्हॉट इज कॉल्ड ॲज अ ग्रोथ, राईट? द ग्रोथ ऑफ द प्लांट इज इनडिटर्मिनेट, रिमेंबर. सो द प्लांट ग्रोथ विच इज देअर ती कशी आहे? इनडिटर्मिनेट, राईट? The plant growth is unique, I tell you, compared to other animals and all. The growth in the plants is unique, right? Because you will find that the plant which is there, it is showing a unlimited growth throughout the life. So as the plant is growing, throughout the life, the growth which is there, it is unlimited. And you know that this is because of what? It is because of what is called as your meristems, right? So you have these meristematic tissue, and just a second. It's not getting rotated. I tried. I don't know what's the matter. Orientation, it says it's locked. I don't know where is the orientation locked. No, I did that. You need to do it later. Rotate the device. No, I tried doing that. It doesn't. Let it be there. And just for this time, we'll do it for the next time, right? No, it doesn't. It is vertical actually. So, anyways, I'm sorry, again, I have to finish this chapter. Vertical or horizontal, let it be. So it is indeterminate, it is unique. The growth is unlimited because of these, what is called as your meristematic tissues. So these meristematic tissues, they are dividing continuously, they are self-perpetuating, right? And you will find that they are dividing, they are producing many cells, and the products which they come out, right? These particular products, they are now not dividing. So these particular products, the new cells which are added, they lose the capacity to divide. So meristematic cells are having capacity to divide. Their products, they lose the capacity to divide. So new cells which are added, right, they are showing the open form of the growth. Remember, the plant growth is open form, it is not closed. The growth in the plants is open, right? So the root and the shoot, the point to be remember here is leading to the elongation. So the meristematic cells, when they are applying, increasing in the shoot, it is increasing in the length, the root is increasing in the length. Whereas here the point which you have to remember is in gymnosperms and in your dicot. So this is what you have to remember, that is in the gymnosperm and it is in the dicot that there is a lateral meristem, right? So there is a lateral meristem, right? And there is what is called as a cambium, which you call as the vascular cambium, and the cork cambium, which is responsible for the increase in the girth. And that particular increase in the girth which is there, that is what is called as the secondary growth. So remember, you have the diagram also where you have a shoot apex, then they have shown a root apex, right? Then you have those cambial, then you have this lateral meristem. So if it is the lateral growth, right, so that lateral growth or increase in the girth is because of this vascular cambium, cork cambium, lateral meristem. And the point to be remembered here is it occurs in gymnosperm and dicot both, right? So it is both gymnosperms as well as dicots which show this what you call as the lateral or the secondary growth. You need to remember that you can measure this particular growth. असं रॅन्डम नाही चाललेलं, आपण ही ग्रोथ काय करू शकतो, मेजर, राईट? सो यू कॅन मेजर हाऊ, यू कॅन मेजर ॲट दी सेल्युलर लेवल. So remember, this growth, it could be measured at the cellular level. You can actually, the basic way of doing is to estimate the increase in the protoplasm. So what you want to find out that how much protoplasm is increased. So that can give you a direct estimate of how much growth happened. म्हणजे मला जर ग्रोथ बघायची किती झाली तर मी काय चेक करणार, किती प्रोटोप्लाझम वाढला. जेवढा प्रोटोप्लाझम वाढला तेवढी काय वाढली आहे, ग्रोथ. But now I cannot directly measure protoplasm. So I will use something else. What will I use? So here what they have told, you can measure a quantity which is directly proportional. So measure a quantity, lakshat gya, which is directly proportional to the increase in the protoplasm. So what is that? It is the increase in the length, increase in the dry weight, increase in the fresh weight, right? Increase in the volume, increase in the number, right? All this can be taken as the measure of your growth, right? One example which they have given, that you have a single maize cell. Please remember this: a single maize apical root meristem. Root apical meristem will multiply and produce 17,500 cells in 1 hour. So one maize cell, how many cells? 17,500. One watermelon cell, right? They have told it will increase in size in around 3,50,000. So here you are actually estimating the number, right? So here what are you estimating? It is the number. Yahan pe you are estimating the number, whereas here you are estimating the size. So at one place we are looking at the number, whereas at other place we are looking at the size. So like if I am measuring the length of pollen tube, I am measuring the growth. But if I am talking about a leaf, I will be more interested in the surface area. So if it is a pollen tube, remember, it is I am measuring the length, and if it is the leaf, it is the surface area, right? So the growth which is there, as I told, that it is measurable. So if it is measurable, you can measure, and what is directly proportional is your protoplasm. What I want you to remember here is that it is fundamental, it is complex, it is permanent, right, thing which is happening, and is increasing, and the gymnosperms and your dicots, they are showing what is called as a secondary growth, right? Then if you talk about the phases, of growth through which this plant grows. You have different phases. You have a meristematic phase. The first phase which is there, that is the meristematic phase. The second phase which is there, that is the elongation phase. And third is what is called as the maturation phase. So these are the three phases of the growth. So all plants will go through these three phases, right? So first is the meristematic phase, where you'll find that the cells are just increasing in number. In elongation, you'll find there is a lot of vacuolation. New cell walls are being deposited. Each cell is increasing in size. So that is elongation. And then there is maturation, when they decide that now they are not going to elongate, but they'll just bring some changes in them. Like they will change their cell wall, they'll change their protoplasm, something or the other they will do. So that you can see in a typical root or shoot apex. So phases, as I told, you can typically see with the first phase in the root and the shoot apex, where you show the meristematic region where the cells are being added. So here you will find that the protoplasm is increasing. I only told that the growth when happens, what first increases is the protoplasm, right? Here you will find that there is a large and a conspicuous nucleus. Don't forget this, that it has a large and a conspicuous what? Nucleus, right? The cell wall is primary. Why? Because it is still has to elongate. So secondary cell walls are not yet deposited because it will become tough. So the cell wall is primary. It is thin. It is made of cellulose because it has to undergo the next phase, that is elongation. It has many plasmodesmatal connection. So remember, so when it is still growing, it has a lot of plasmodesmatal connections. So you have to remember this. So in this phase, what you have to remember, it is in the root and the shoot apex. The protoplasm is increasing. It has a large conspicuous nucleus. Cell wall is primary, thin cellulose, and it has many plasmodesmatal connection. Why and all I'm skipping here because you have to like go through one by one. Next to this, remember the word which is proximal. So proximal to it. So if you start from the tip, the first is the meristematic. The proximal to this is your elongation. Now proximal to elongation, you will have maturation. So the above one, like you go away from the tip. So tip में meristematic है, उसके बाद कौन सा है? Elongation. उसके बाद कौन सा है आपका? Maturation. So proximal to that is elongation, right? And then I told after the elongation you have the next phase, that is what is called as the maturation. So maturation I told is characterized by thickenings, various thickenings. मतलब you will find that various layers of cell walls are deposited. You will find that there are a lot of modifications also in the protoplasm depending on what the cell decides to be in future. You will find that will decide what will happen to the cell wall and what will happen to its protoplasm, whether it's going to keep the protoplasm or it's going to lose the protoplasm. Anyways, right? Then you have what is called as the growth rate, right? The rate at which it is increasing. So as the growth increases with time, right, we say that the growth rate is high, right? So time is important. 10 days approximately remaining. So now at what rate you are studying is more important. So growth rate is like at what rate the growth is taking place. We basically define this growth rate into two. You have an arithmetic growth and then you have what is called as a geometric growth. In arithmetic growth, when a cell undergoes mitosis and forms two cells, you will find that out of these two cells, only one cell it divides, the other doesn't divide. So only one cell when it divides, other does not divide, then that is called as the arithmetic growth. If you plot the graph of this growth, remember here you're plotting the time, you're plotting the size, length, whatever it is a straight line. So one cell divides, other cells differentiates and matures. That is what is called as the arithmetic growth phase. Here you have to remember this is shown by elongating root at a constant rate. हे लक्षात ठेवायचं आपल्याला की एक्झाम्पल इथं काय दिलंय, root elongating at the constant rate. That is the example of which growth? Arithmetic growth, right? So here they have told it is LT is equal to L0 plus RT. So LT is your final length, L0 is your length when you actually started the experiment, R is your growth rate, the intensity growth rate, and T is the time for which you are studying the growth, right? So they can you may have some questions, like some mathematical questions on this, like for a particular plant, if R is say 5 units per day, right? So, and the plant is growing for 10 days, right? The initial growth of the plant or length of the plant was 20 units. So find the length after 10 days. So length after 10 days would be like L0, 20 units, whatever it is. R, that is your 5, and T, that is your 10. So it is 20 plus 50, that is equal to 70. So like this, or they may also ask you to calculate R by giving you all other values. So you should be ready with this. So LT is the final growth, L0 is the initial growth, whatever the length, size, weight, whatever it is. R is the growth rate and T is your time. And graph is कैसा line मैंने बोला आपको, straight line. Example हमने कौन सा बोला, root elongating at the constant rate, right? Then you have another particular growth which is called as the geometric growth, right? Or here all the cells are dividing, there is no differentiation. Matlab you have one cell and that cell it thinks that I should keep on dividing, I should keep on dividing. So एक के चार नहीं होते, एक के दो होंगे, फिर दो के चार होंगे, चार के आठ होंगे, आठ के 16 होंगे. So all cells are dividing geometrically, they are dividing. So the graph which you get here is sigmoid or S shaped. So the phases which you have here is your lag, log, and stationary. So you have a S-shaped growth here. And the equation here which you have is W1 is equal to W0 into e raised to RT. Right. So here also you have the size or the weight of the organism. Here you have the time. So W1 is the weight after time, a particular period. W0 is the initial weight. E is the natural base of logarithm, and R is your growth rate, and T is your time, right? Time of whatever growth, whatever it took place. So it is S shape. Remember that living organism in natural environments, all cells and tissues of your organs, right? They all show this phase. That is what is called as your sigmoidal shape growth curve. जसं तिथं अरिथमेटिकचं एक्झाम्पल आपण काय लक्षात ठेवलं, रूट elongating at the constant rate. तसं इथं all organisms, all living organisms which are living in the natural environment, all cells, tissues and organs of the plant, they all are showing which shaped curve? S-shaped curve, that is sigmoid shaped curve. R, I told, R is what? Your relative growth rate. R is referred to as what is called as relative growth rate. I'm carrying this so that I don't want to add any extra word. I don't want to miss out any word. So I want to go one by one what is given in the NCERT. So R is your relative growth rate, ability of the plant to form new plant material, and that they have referred to as efficiency index. So point to be remembered here is that it is the efficiency index. So R is the intrinsic growth rate, R is the ability of the plant to form a new plant material, R is the relative growth rate, and R is what is called as your efficiency index. Here they have told W1 depends on W0. How much, with how much you have started, that will decide how exponentially you will go. आपने 2 साल कितना पढ़ा, that will decide कि अभी ये 10 दिन में क्या होने वाला है, right? So what is your initial knowledge? What is the initial growth? If I have to start with, I have 100 cells, then obviously I will reach a higher target. So remember, in assertion reason, W1 depends on W0. W1 also equally depends on the R. Also, it equally depends on the T. So all these factors, what remains constant is your E, that is your base of your natural logarithm, right? So that is regarding the arithmetic and the geometric. So points to remember, LT is equal to L0 plus RT, a straight line graph, a constant growth rate, W1 is equal to W0 into e raised to RT, a sigmoid shaped graph, root elongating at a constant rate, and all organism plants living in the natural environments and all plant parts are showing which shape growth? S shape, right? If you're talking about measuring this growth, so you can have a quantitative comparison. I request you to look at the images in your NCERT related to the arithmetic growth, geometric growth. So they have shown how arithmetic growth takes place, how geometric growth is taking place. They have also shown that how when a zygote divides, the first phase is the geometric phase, and then when it divides, the next phase is the arithmetic phase. Last year in NCERT, NEET, this question was there, identify A and B. So it was the geometric growth and it was the arithmetic growth. So similarly, the diagrams of geometric and arithmetic also, where only one cell is dividing and both cells are dividing, keep a watch to that also. If you are measuring or comparing of total growth, right, per unit time, then they have told that that is the absolute growth. उस growth को उन्होंने कौन सी growth बोले हैं? Absolute growth. So absolute growth, I told, when you are measuring the total growth, whatever growth took place, that is absolute growth. Now here point to remember is that growth of given system per unit time, the word to remember is on common basis per unit time of initial parameter. You remember those two leaves, one going from 5 to 10 and other going from 50 to 55. So in both the absolute growth is 5, remember. But relative growth, how we calculated? This was 5 out of 5 and this was 5 out of 50. So if you are taking into account the initial parameter, so growth on common basis per unit time based on the initial parameter. So here it was like 5 into 5 into 100. I have 100% of growth, and here I have 10% of growth. So relative growth, remember both the leaves A and B will have same absolute growth, but relative growth is more in A. So if I am taking the growth as a common basis of the initial parameter, then that growth is called as which growth? Relative growth. But if it is only a total growth per unit time, right, then that is what is called as the absolute growth. But when I am comparing it to the initial parameter, it is the relative growth. In these two leaves, one going from 5 to 10 was having more relative growth. One going from 50 to 55 was having less relative growth, right? The various conditions that you require for growth. Remember, you require various conditions for growth. Some of the conditions which they have mentioned is your water, then your oxygen, then your nutrients. So when a cell is enlarging, when we say that there is elongation phase, so enlargement of that cell is because of the water, right? Turgidity, extension is because of the water. Everything, every metabolic activity you can link to water. Enzymes which are there, they are also active in the aqueous medium. So water is essential for growth. Oxygen you require for metabolic energy, because aerobic respiration is going to require oxygen. And nutrients, you know, whether it is a macronutrient or whether it is a micronutrient, it is essential for synthesizing protoplasm, and it is essential for providing what is called as the energy, right? So that particular growth, right, what we call as, so that is water, oxygen and nutrients. So as I told, water, turgidity, elongation, extension, aqueous medium for the enzymes, oxygen, metabolic energy, respiration, and nutrient, macro and micro for synthesizing the protoplasm, right? Optimum temperature is also important, and if you are going like away from the optimum, if the temperatures are going very less or the temperatures are going very high, that may be detrimental. It may even lead to the death. So variations are not very good. We all don't like much variations, right? Other things which are required for growth, remember they have clearly mentioned, you require light, you require gravity, and the point to remember here is that they have told that this is required at certain conditions. So under certain what you call as stages of the growth, what you require is light and gravity. So all these factors which are there, so they define what is called as your growth. Now you come to those three magical words. The differentiation, the de-differentiation and the redifferentiation. So it is differentiation, then you have de-differentiation, then you have redifferentiation, and you are in love with these words. Right? So when you start with a meristematic cell, right, and the cell is like growing, multiplying, and at one point it decides that now I'm not going to multiply. It shows a change in the protoplasm. What it tries to do is it tries to differentiate itself. It tries to become a trackery element. It tries to deposit that protoplasm. It tries to make the cell walls lignified so that it can perform the proper function of carrying the water. So when the cell was dividing, right, at one point it stops dividing, and then it decides to become a sclerenchyma, parenchyma, collenchyma, or whatever it loves to be. That is differentiation. That is the first step. That is what is called as what? Differentiation. So When you start from a meristematic cell and then stop dividing and become mature, that is differentiation. When a mature cell which has lost the capacity to divide decides to divide again, now this cell, it thinks like meristematic cells is dividing, why I'm not dividing, so this starts dividing again, so that is de-differentiation. Remember, your cambium, the vascular cambium, the cork cambium, they are the product of de-differentiation. Your primary tissues, they are differentiation. Your cambium, vascular and cork and intravascular, that is de-differentiation. When they differentiate, after dividing again, they again differentiate. So now they are differentiating for the second time. So that is re-differentiation. So all the secondary tissues in the plant, your secondary phloem, secondary xylem, all that are the products of what is called as your re-differentiation. So when you divide, stop, differentiation. Stop, again divide, de-differentiation. Again stop, re-differentiation. Cambium, de-differentiation, primary tissues this, secondary tissues is this. Now, what will happen to the cell will depend on where this cell is located. Like if you take a particular cell away from the root, like, and put it at the tip, it probably will become the part of root cap. And if you take this cell and put it towards the periphery, probably it will become the part of the epidermis. So location of that particular cell is going to decide the fate of the cell. So that is what we call as regarding your growth. So let us see what they have to say. The root and the shoot apical meristems, they differentiate, they mature, they perform specific function, show major structural changes where two places, cell wall and protoplasm. Example they have shown of these trackery elements where the cell, it loses the protoplasm. You have a strong elastic lignocellulosic cell wall. The water which is there, it is to be carried at long distances under pressure, and these cells now have lost the capacity to divide. That is differentiation. And these cells which have lost the capacity to divide again divide, that is de-differentiation. Intervascular cambium and cork cambium, they form because of de-differentiation. These cells divide, again differentiate, they again lose the capacity to divide. That is what is called as what? Re-differentiation, right? So this is how it takes place. Here you have to remember that the growth which is there, it is of the open form, it is indeterminate or determinate both, and you have to remember that differentiation is also open. So see, growth was open, here differentiation is also open. Differentiation क्यों open है? इन्होंने बोला है because from the same meristem you can give rise to different types of tissues. So that is open. Like हम बोलते हैं ना, the session is open for discussions. You can ask anything. Like your parents say that we are open, she can do anything or he can do anything. So the meristem also, when it is forming the cells, it says, okay, I'm just forming, you decide what you want to do. So they are open. So differentiation is also open, growth is also open in plants. And I told that what structure will be at the maturity will depend on the place. If it is away at the tip, it will form a root cap. If it is at the periphery, it will form the epidermis, right? I told some of all these processes is what is called as development, right? From the point where the cell is being formed and it is being growing and maturing, and all the changes which is taking place in the life of that particular plant is called as development. Like humans also. Okay, let us not go into that satsang thing, but development, as I told, like whatever changes are taking place. See, plant is not only continuously growing and multiplying, it is also becoming old, it is also becoming mature. So including all that, so different pathways which the plant is following, different environmental and phases in life which the plant is going through, that is what is called as development. If you see, they have given that chart, right, where you start from a meristematic cell, then there is a plasmatic growth, then there is maturation, then there is senescence, then there is death. So here sometimes from plasmatic growth you come back here, right? So here it is what you call as cell division. After the meristematic growth, then you have plasmatic growth, then you have maturation, then you have senescence, then you have death. And here sometimes the cell is growing back after the plasmatic growth without maturation. So here you have increase in the number, here you have extension. So all that you have to remember, that chart, right? Please have a look at it in your NCERT. So development, as I told, it includes everything. Plants, they behave differently. in different environment and even during different phases of life, and that is what is called as plasticity. So plants producing different types of leaves under different conditions is called as heterophyly. If the plant is producing different types of leaves, that is what is called as heterophyly. It is producing different leaves under juvenile and adult phase. So if you look at the adult plant and if you look at the juvenile plant, the leaves, they are different. Which are the examples which they have given? Cotton, coriander and larkspur. So they show juvenile and adult heterophyly. So at different stages they are different. Whereas heterophyly in air and water, example they have given now buttercup. I request you to please look at those diagrams given in the textbook, right, of those leaves, because they may show you those leaves and they may ask you, "ये किसका है? ये buttercup का है? आपको कोथंबीर लगेंगी," right? But whether it is of buttercup or coriander or larkspur, right? So they are given, I think, the example of larkspur and buttercup as far as the leaves are concerned. So they have shown those leaves, right? They have shown, right? So the juvenile and adult, as I told, that is heterophyly. In cotton, coriander and larkspur, whereas in air and water it is in the buttercup, right? So that is what is called as differentiation, right? Plasticity actually. So growth and differentiation, they have told that they are related. Growth, differentiation, development, all are closely related. 100 times they have told development is the sum of growth and differentiation taking place because of both intrinsic factors. Intrinsic में उन्होंने बोला है there are intracellular. So you have some intrinsic may you have intracellular inside the cell, that is genetic. Some they have told intercellular, that is your plant growth factors, plant growth regulators, the auxins, cytokinins, which we are going to study. And you have some external factors, that is your light, then your temperature, then your oxygen, then your water, then your nutrition. Oh my God. So all this is affecting the growth. आपल्याला वाटते is a plant growth खूप सोपी आहे, but it depends on, remember, intrinsic मध्ये दोन, एक इंट्रासेल्युलर, दुसरा इंटरसेल्युलर. इंट्रासेल्युलर मध्ये काय सांगितलंय त्यांनी? जेनेटिक. आणि इंटरसेल्युलर मध्ये काय सांगितलंय त्यांनी? Yes, intercellular, what they have told, the plant growth regulators, external, the light, the temperature, the oxygen, the water and the nutrition, right? So all these are going to affect what we call as the growth of the plant. Please do not ignore all this one word, one line, because it really matters, because I'm sure maybe this time you're going to have 4-5 questions from this. That's what I guess. I don't know, but now coming to your most important topic. Oh my God, it's around 4:46. We have only 15-20 minutes left, and you don't know why I'm in a hurry. I have a class and there is one more problem. So now, yes, you're saying something. So PGR, the basic thing which we have to, like, which you all love probably, is your PGR, the plant growth regulators. Remember, these plant growth regulators, they are simple but they are diverse. Small molecules, simple, diverse. What you have to remember is some of them, they are indole compounds, and indole compounds में कौन है आपका? IAA, indole acetic acid. So they are derivatives of indole. Some of them are derivatives of your adenine. You remember this N6-furfuryl amino purine. So you have this N6-furfuryl. Please remember this. They may ask you and match the following, right? So adenine derivatives, that is your N6-furfuryl amino purine. You know that it is your kinetin, the cytokinin thing. Then some of them, they are derivatives of your carotenoid derivative. You know your ABA, that is your abscisic acid, right? Which is a, anyways. Then you have some which are derivatives of terpenes, that is your gibberellic acid, and some you have which are gaseous, that is your ethylene, right? So these are, you can say, plant growth regulators, alternative names. I don't know why they have so many names in biology, but then you have growth substances, hormones, plant growth hormones or phytohormones. These are again categorized into two. One are growth promoting. So some are growth promoting, which